Okay, so hello everyone, and I'm very pleased that we have Sandy Hetherington here today to deliver his research seminar. And Sandy is also the exceptional lecturer of the Paleontological Association. So that's an award, I would say, that is awarded once a year. And the exceptional lecturer delivers the Innovations in Paleontology lecture series. So we're in line with, I think, a total of 5 or 6 institutions. And this is his second talk. The first one was to the Geological Society of London. And his talk will explore land-plant evolution. But let me, before I say more about the talk, let me give a bit of background. So Sandy is actually a geologist by training. He got an MSci from Bristol in geology. So that's really where he's coming from, but he is ultimately interdisciplinary, and his PhD, and I'm reading from his biography, is a interdisciplinary biosciences is the first one I came across with that title, but absolutely justified because he combines paleontology with genetic research on plants, and has created some really thought-provoking advances on the field of plant evolution. So he finished his PhD in 2017. And stayed, and that was in Oxford, and stayed in Oxford for a little bit longer, but has just been awarded one of the Future Leaders Research Fellowships, and took that to move to Edinburgh, I think just in October, you said, right? So
yeah, just
in the middle of the pandemic, rather interesting times to move across the country. But he's starting to establish himself there now, and will take on a permanent position there at the end of that leadership award. And today he will talk to us about In the Search of the Roots of Roots, 400 million years of plant evolution. Okay, I take over, please.
Brilliant. Well, thank you, Yves, for that introduction. I'll just share my screen, and if you could just give me a nod to let me know you can see that okay?
Yep.
Brilliant. Just get this. Fantastic. Brilliant. Well, thank you so much for the invitation to be here today. And I just really want to start off by saying a special thank you to the Paleontological Association for really giving me the opportunity to present this lecture as part of the Innovations in Paleontology lecture series. So let's get started. So I really wanted to start this lecture off with a photograph. So this is a photograph by the amazing photojournalist Emmanuel Rondeau. And Emmanuel said, allowed me to include this photo in this presentation today. So what I'd like you to do is to just take a few seconds, 30 seconds or so, just to really study this picture in detail. So I remember seeing this picture for the first time at the Wildlife Photographer of the Year exhibition in London in 2019. And I was really drawn to this picture because of this rich rainforest ecosystem. But I remember when I was examining it and I got closer to it, that I suddenly came across the jaguar that I'm sure many of you have seen peeping its head round the fallen tree stump here. And it was as soon as I'd seen this jaguar that really the whole picture seemed to change. This rich rainforest ecosystem just seemed to blend into the background, and all I could really see and focus on was looking into the eyes of this jaguar. And I found this a really amazing example about how our brains are adapted to be able to see and recognize animals in ecosystems. Whether those animals are predators or prey, whether we're recognizing them from their color or their movement, it's really remarkable how we're able to identify these animals. But really the flip side of this is that more generally, it often means that the plants in the ecosystem therefore just blur into the background and become the backdrop of the image. Now, this is part of what we call the wider phenomena of plant blindness, where plants often go underappreciated or unnoticed in an ecosystem. And this is not just prevalent when we're thinking about living species, this is also present when we're thinking about fossil species too. Now, to illustrate this, here are two fantastic paleo reconstructions by the paleoartist Richard Bisley. Now, I think these paleo reconstructions are absolutely fantastic at bringing these ancient ecosystems back to life from the Jurassic period and the Carboniferous. And despite the fact that I'm fascinated by plant evolution and work on fossil plants, I really can't help myself from my eyes being drawn to these charismatic animals right at the front of shots, such as the dinosaur or these giant insects. And in both cases, again, these plants kind of blend into the background as these forest backdrops. And this goes the whole way back, even further back into the geological record, even when we're thinking about some of our earliest ancestors coming onto land. Even when we're picturing them coming onto land, they're coming onto an ecosystem which is already covered by this rich forest. And really, the problem with this is that we begin to think of this forest ecosystem as simply being a static backdrop to animal evolution. And we often think that this forest was just there and has been there for the entire time of animal evolution. But by doing this, we're missing a very interesting aspect of plant evolution that's going on at the same time. And just to illustrate this, if we just look at each one of these forest ecosystems here, despite the fact they might superficially look quite similar, they're fundamentally different. In the present ecosystem, this rainforest, the trees there are pretty much all flowering plant trees, angiosperms. And angiosperms were basically only in their very infancy or hadn't actually evolved yet in the Jurassic when this dinosaur was roaming the ecosystem. Instead, these forests were formed by seed plants or gymnosperm trees. And as we go back further into the geological record, these forests get stranger and more distinct from any forest alive today. Take 310 million years ago in the Carboniferous, you can see these giant trees there. Most of these trees are in fact giant ferns and club mosses and horsetails, which today are just small herbaceous plants. And finally, when we go back to the Devonian 375 million years ago, the trees in these forests have no living analogues today, and they're actually all of major groups which have now gone extinct. And furthermore, plant evolution didn't just begin when our ancestors came onto land. There's actually a rich record of fossil plants going back to 470 million years. So 470 million years ago, we find the first records such as these tiny spores that plants were on land. And in between this time and the late Devonian, there's best part of 100 million years of evolution where plants were colonizing the land and completely transforming it, such as these early Cooksonia species in the Silurian and onto these rich kind of herbaceous ecosystems in the early Devonian. So really, land plants really did pave the way for the diversification of animals on land. And so for me, I really think plants are well worth studying in their own right. They have a fascinating evolutionary history. And for me, my particular interest is in this period of time here of how plants went from these earliest terrestrial ecosystems in the Silurian and Ordovician to these first forests in the late Devonian. So what key innovations enable plants to diversify to produce the forests we're so familiar familiar with seeing today? And in my opinion, one of the innovations that enable plants to undergo this radiation was the evolution of plant roots. Now, plant roots are things that you might have not thought about that much because they're typically the hidden half of plants that are below ground. But again, in my opinion, I think these roots have actually had a profound influence on the terrestrial surface. And to illustrate this, I really think plant roots change the Earth both biologically and environmentally. So biologically, the evolution of roots enable plants to enormously increase their rooting function of anchorage combined with nutrient uptake and transport. And this is really well illustrated in this diagram here of what's going on in rooting system evolution during the Devonian period, roughly 400 million years ago. As you can see, as rooting systems are becoming deeper and more complex, they're being mirrored above ground by an increase in shoot complexity, leading for the first time to the evolution of the trees in the mid-Devonian, and then onto these giant trees in the Carboniferous. So I think there's no doubt that the evolution of roots played a key role biologically. but they also played a key role environmentally. Roots led for the first time to the evolution of deep soils, and they also changed fluvial systems. And the way they did that is roots bound together soil particles, canalizing water flow to produce these characteristic meandering river channels we're so familiar with on the terrestrial surface today. And finally, despite being in the ground, they really did change the composition of the atmosphere. Now, to illustrate this, here's a graph of atmospheric CO2 through geological time. So we've got CO2 in parts per million and geological time on here with 500 million years ago through to the present day. And in this blue box here, I've highlighted the period in time when roots are really exploding in their diversity. Now, as you can see, this is correlated with an enormous decrease in atmospheric CO2, shown in this black line and the gray bar here. And we now know that this is not simply correlation, it's in fact causation. And that's because roots were increasing the weathering of silicate rocks, drawing down atmospheric CO2 and reducing atmospheric CO2 rates. So roots, despite being in the ground, had this profound impact on the atmosphere. So just in these two slides, I really hope I've given a bit of a pitch for why I think the evolution of roots has been crucial for all life on Earth. And over the next 30 or 40 minutes or so, what I'm going to do is to take you on a bit of a journey to try and get to the root of roots. And to do that, we're going to be focusing in on one plant group known as the lycophytes. So the lycophytes are the earliest diverging lineage of the vascular plants, and they're shown here in this blue box here. So what I'm showing on this slide is what's known as a family tree or a phylogeny of living land plant species today. And living land plant species today can be separated into two major groups. Over here in green, we have the non-vascular plants. These are the hornworts, the mosses, and the liverworts. And they sit as sister to this much larger group of plants in orange, known as the vascular plants. Now, the vascular plants are named because they have specialized conducting tissue, water-conducting xylem, and food or sugar-conducting phloem. And for me, the lycophytes hold this key position because they're the earliest diverging branch of this key group, the vascular plants. And the lycophytes today are actually split into three major groups, the Lycopodiales, Selaginales, and the Isoetales. And here are photographs of examples of each of these major groups in here. Now you can see that these plants look, their shoot systems look very different, and they're also in very different ecological settings. This is an alpine species, and this species of Selaginella you often find crawling on the jungle's floors, and this Isoetes species is an aquatic species. So they look different, they're in different ecological settings. But something I find absolutely fascinating about these plants is if we turn to their rooting systems, we find that they look almost identical. And they have a type of architecture we call dichotomous branching architecture. This is where a single axis grows and it splits in two and it splits in two again. So despite these differences in ecology, their roots look remarkably similar. And it's not just the overall architecture that looks similar. When we zoom in more detail, we find that in actual fact, they all have the same overall characteristic anatomy. And so, for example, if we take one of these roots and we zoom in on the very tip, and we examine that under the scanning electron microscope, these are the results we see. So this region you can see in this box here is the apex of the root, which we call the root meristem. So that meristem contains a self-renewing population of stem cells, which enable the root to continue growing through its lifespan. And in false color, in yellow here, I've highlighted one very key tissue. This is the sole-defining feature of roots today, and it's known as the root cap. You can see it's forming a cap-like tissue over the apex and has a key role in protecting those stem cells underneath and helping that root grow through the soil environment. And the other key anatomical feature we find on all the roots of these species are the presence of root hairs. Now you can see these hair-like projections coming from the surface of the root, and the surface of the root is known as the epidermis, and these are root hairs. They're cells which enormously increase the surface area for the plant roots. And again, a root meristem with a root cap and root hairs is a defining feature of all of these groups of roots. And finally, it's not just the architecture and the anatomy, which is similar, but it's also the development of these rooting systems, which is conserved. So what I'm showing over here on the left is a cartoon of these different types of architecture. This is the dichotomous rooting architecture I introduced on the previous slide. And this is actually formed by development from that meristem in a very specific way. So here's a 3 cartoons as a kind of time lapse through how dichotomous branching works. This first cartoon just shows a normal root meristem like the one I showed you on the previous slide. And as it grows, it physically splits in two to produce 2 daughter roots that are produced. And I always find it easier to picture this by watching a video. So here's a great video by Fang et al. I'd like you to focus in down the base here on this root as it's growing, it's elongating, enlarging slightly, and finally actually splits in 2. And again, the same thing's happening to this root up in the top corner as well. So from studying the roots of living lycophytes, what we can conclude is they have a remarkably conserved architecture, anatomy, and development. And as someone who's really fascinated by evolution, this instantly conjures up a few really interesting questions that I wanted to get to the bottom of. Given how conserved these are in living species, I wanted to know, are these features also conserved in the fossil relatives of lycophytes? And furthermore, when did these roots evolve? And to do that, we need to leave the living plants behind and turn to the geological record. So we're going to start off our journey in the Carboniferous period. We've gone back 300 million years, and here's a paleo reconstruction of what the Earth looked like 300 million years ago. The continental plates were grouped together into the Pangean supercontinent, and the red star here represents where Britain was just a bit north of the equator in a tropical environment. Now, for me, if I ever want to picture a bit like what the Carboniferous must have been like, I often think it must have been similar to something like a tropical climate fern house that you can visit in a botanic gardens. So one of the great things about being here in Edinburgh is being close to the Royal Botanic Gardens, which has some absolutely fantastic fern houses. And if we go to the fern house here in Edinburgh, we see lycophytes today. And here's an example of one of them. You can see its leafy shoot, and they form these highly branched leafy axes. And what's amazing is that if we actually look at the Carboniferous rocks that are preserved around Edinburgh, we see lycophytes that look almost exactly the same. This is an example in the Hugh Miller collection at the National Museums of Scotland called Lepidodendron elegans. And you can see again, this is the leafy shoot preserved in this black outline here, which is also undergoing the similar branching pattern. But there's one major difference between the lycophytes in these fern houses in the Botanic Gardens today and the lycophytes that were living in Edinburgh 300 million years ago, and that's their scale. So this is that reconstruction I showed you earlier of a Carboniferous coal swamp environment. So I'd like you to take your eyes away from those characteristic animals, but this time think a bit about these giant trees. And each one of these giant trees is actually a giant tree lycophyte, or an arborescent lycopsid. These trees, despite the fact that their living relatives only grow to a few centimeters tall today, could reach enormous heights of 50 meters. And they were key components of the Carboniferous coal swamp forests, which produce lots of the coal we burn today. And because they were so frequently preserved with the coal that they preserved, we've actually got a good understanding of these trees and a great fossil record, including a fossil record of their rooting systems. Now, the rooting systems of these trees go by the name Stigmaria ficoides, and they're some of the most abundant of these large tree root fossils we find during the Carboniferous. And some of their most iconic fossil sites are sites such as Fossil Grove, which is right in the centre of Glasgow, where they've discovered a fossil forest of these trees preserved in situ. So you can see one of these giant tree stumps and the rooting axes radiating out from the base. But they've cropped up all over the world, and here's an enormous example from Osnabrück in Germany, showing just how big these rooting systems could get. Because these rooting systems are so big and so abundant, we've actually we thought we had a relatively good understanding of the structure of these trees going back to the early 19th century. So here are some illustrations of these rooting systems from the early 19th century, and you can see that they overall have a very similar pattern. So let's take an example from Charles Lyell, one of the godfathers of geology, his own geological notebook. So here's one of his illustrations of stigmaria. So you can see at the center, it has this woody axis with these pot marks on, and then radiating out the side from this axis are these tapered structures called rootlets. Now you can see this same overall pattern has also been shown in Binney, and even if when we skip forward over 150 years to a much more recent reconstruction, we can see again that these large axes had a large woody axis at the center and these small tapered rootlets radiating out. And you can see that there's clearly in all of these reconstructions no real evidence that these rootlets were branching. And furthermore, when we find these fossils preserved with exceptional preservation, there is no clear evidence that they developed root hairs. So here's an example of one of these stigmarian rootlets preserved with exceptional preservation from the Carboniferous. This is a thin section looked at under the microscope, and you can see this is what happens if we cut a transverse section through one of those rootlets. You can see the outline of the axis here, and you can see this is the surface layer. There's no evidence of those hair-like projections from the surface. So at this point, what we can see when we look at these giant trees is their rooting system looks fundamentally different to the roots of living lycophytes today, including the roots of their closest living relative, plants of the genus Isoetes. And if you remember, I've described to you the roots of Isoetes earlier. They conform to normal lycophyte roots in that they're dichotomously branched. And when we look at the surface, we see evidence of root hairs developing from their epidermal surface. So at this point, you can see that there's clearly this very big difference, we were thought that a very big difference between the roots of living Isoetes and these giant tree Lycopsids. And in fact, this difference was so big that people predicted that the roots of these giant trees were not roots at all, but were in fact modified leaves. And the reason for that is they saw similarities in the structure of these rootlets with the leaves of the above ground parts of the plant. So here's that reconstruction from Binney showing the rooting system below ground. And this is a reconstruction of the above ground part. And you can clearly see both are covered in these tapered projections called rootlets below ground and leaves above ground. So this has been put forward as the modified shoot hypothesis. Now, despite the fact this has its advantages for trying to understand these early rooting systems, for me, after spending some time examining the roots of living lycophytes, it really came as quite a shock because, as I said earlier, the roots of lycophytes today are just so highly conserved despite their different ecologies. And so I began to wonder, is it possible that these rooting systems have simply been misinterpreted? And really, the eureka moment came in this project when I was visiting Dr. Chris Berry at the University of Cardiff and looking through some of his thin section collections that they had in Cardiff. And it was while doing that that I came across this specimen here in the red box. This is another transverse section through a root of a Carboniferous tree Lycopsid. But what was very special about this fossil is that when we examined the epidermal surface, you could see clear evidence of these root hairs developing from the epidermal surface. And for comparison, you can see these look almost identical to the root hairs you see in living Isoetes today. So this was the first line of evidence to suggest that these rootlets and these tree Lycopsids were actually more similar than we previously thought to the roots of living Isoetes. And so at this point, after finding root hairs, the next question was, well, did these roots also branch? And so to examine this, I examined the amazing collections that they have in the British Geological Survey. And it was while examining those collections that I came across these two remarkable fossils. So these are two highly branched stigmarian rootlets with little Isoetes roots for scale. So you can see that despite the fact these fossil roots are much, much larger than the Isoetes roots, they have the same overall architecture. So at this point, we've been able to find evidence of root hairs, and we found evidence of branching. However, just finding two isolated examples of branching is never really enough to convince you that these roots were highly branched like the living Isoetes roots are today. And so what we really needed to do was to begin to think a bit more generally about whether it was possible that these roots were highly branched and whether these highly branched roots could have potentially been missed in the fossil record. And so we turned again to those artist reconstructions of what these trees look like. We knew they had these large woody axes with these rootlets radiating out the side. But again, when you look in collections, you realize that actually most of this, quite a bit of this is actually artistic license, because rarely do we find an example where you can actually trace the rootlets from the woody part all the way to their tips. So to illustrate this, here's a great specimen in the Natural History Museum in London. You can see the large woody axis at the center and radiating out of these rootlets. But you can see that none of these rootlets are actually terminating, and they would have stretched off into the surrounding matrix. So we predicted that potentially, was it possible that in fact they were branching, they were just branching further away from the woody axis. Now, to investigate this hypothesis, what we needed was to examine lots and lots of fossil soil. And we're actually very lucky in the Carboniferous to have large amounts of fossil soil preserved as fossil deposits known as coal balls. So here's an example of a coal ball from the Manchester Museum. Now, if you take a coal ball and you cut it into a very thin slice, a thin section, you see this is the result you get. And you can examine that under a microscope and see the amazing preservation of the plant tissues preserved in this PT fossil soil. You can see here. Now, for me, this fossil soil was so exciting because it was very easy to identify the roots of these giant tree lycophytes. And that's because they had a very characteristic anatomy. So you can see an example of one here. You see this large outline, large circular outline, and then there's a large airspace in the middle before a central vascular strand. Now, this is a very diagnostic type of anatomy, and therefore meant it was nice and easy to spot lots of these rootlets in this fossil soil. So each one of these white arrowheads indicates one of these fossil rootlets. So therefore, what we could do is it meant very quickly, we could go and examine the structure and especially the diameter of lots of these fossil rootlets. So at this point here, we had a way of measuring large numbers of the diameters of fossil rootlets, but how could we compare them between either being tapered or being branched? Now, to do that, we made a very simple prediction about predicting the frequency of finding a rootlet that was tapered, which is what people previously predicted, or if those rootlets were branched as we predicted. And the results of this very simple model are shown on the right-hand side in this frequency histogram. So we got rootlet diameter along the base, so small diameter roots and large diameter roots, and then the frequency of finding them. So let's start with the tapered model. So the tapered model suggests that if I'm looking through fossil soil, I would be just as likely to find the large diameter part at the top as the small diameter part at the bottom. So therefore it has the same frequency of finding it in fossil soil. By contrast, in the case of the branch root, as the root branches, it decreases in size, but as it decreases in size, it increases in number. So one large axis produces 2 and then 4, and gradually you enormously increase the number of these small terminal rootlets in a branch rootlet. And therefore, if we were looking for them in fossil soil, we would therefore be far more likely to find these small diameter rootlets than large diameter rootlets. So at this point, what I needed to do was to test the hypothesis of the model. And so I turned to these coal ball fossils, and I measured the diameter of almost 800 stigmarian rootlets preserved in coal balls. And the results of doing that are shown here in black in this frequency histogram. And what you can see is, and I was really surprised to see this myself, is it seems to mirror almost perfectly what we predicted with our branch root model. So at this point here, we took together the evidence of root hairs, of branching based on compression fossils, and branching based on this new coal ball data. And with the help of the amazing botanical artist Rosemary Wise, we put forward this new reconstruction for what these tree lycophytes look like. So you can see that large woody axis at the base here, and then radiating out on all sides with just vast numbers of roots. We predicted up to 26,000 roots per meter of rooting axis. And then these rootlets that radiate from the sides, they were highly branched, and in their terminal regions, they produced root hairs. And this in turn produced a surface area over 5 times larger than what people previously thought for these giant trees. And furthermore, because of sites like Fossil Grove, we could actually zoom out and predict what the entire ecosystem would have looked like. And in this case, because we know the density these trees grew at, we can predict that their rooting systems form these vast overlapping root plates, which would have anchored these trees to the substrate. Now, at this point here, when you have a model like this, what you really want to do is to turn back to the rock record to test the hypotheses. So to do that, we had an excursion to look at fossil rooting systems preserved on the Fife Coast, just a bit north of Edinburgh. And what we can see here is these large carboniferous beds exposed on the seafront. And what's amazing is that if you just walk along the coast there, you can really find these enormous rooting axes just weathering out. So you can see this large woody rooting axis weathering out of this very clear, pale sandstone. And you can also see these dark outlines of these rootlets which were connected there. But obviously you can only trace them for a few centimeters. However, what was great is there was also another mode of preservation we found there, where rather than just weathering out, the large woody axes and the rootlets were preserved as these inky blue outlines in the sandstone. So you can see these rootlets, these little circles in transverse section, as they're growing out into the sediment. So this sandstone was a great way for us to think about and imagine the overall rootlet network in total. So based on our prediction of our new model, we predicted that as we move further from the woody part, we'd actually find masses of these root hairs, these rootlets, preserved in the sandstone. And remarkably, this is exactly what we found. So what we're looking at here is that very pure, creamy sandstone. But as you can see, the whole surface has this mottled appearance, this kind of pockmarked appearance, and each one of these are actually the marks of these fossil rooting systems within this sandstone. And we can zoom in on these in a bit more detail. When you do that, you can see clear evidence of circular outlines of these rootlets at a variety of different diameters, just like we predicted in our model. And the main thing is we can just see evidence of rootlets absolutely everywhere in this fossil soil. So really, this was able to help us verify this model for what the rooting systems of these giant trees look like. Now, for me, this was particularly important because it told us that the rooting system of these giant trees was similar to the roots of living lycophytes today. So there was no need to interpret their rooting systems as modified leaves. Instead, we predicted that their roots were in fact true roots like that of other lycophytes. So at this point, we've been able to show that their structure had been conserved. But a real dream is then to try and predict if their structure had been conserved. Does that mean that the genes that control them are also conserved? Now, in the ideal world, what I'd love to be able to do is just turn to these fossils and be able to extract them, take them back to the lab and extract DNA and RNA from them. But despite what some filmmakers might have you believe, it's just not possible to get DNA and RNA out of specimens much older than a million years, let alone 300 million years. But that doesn't mean we can't predict what genes were present there. And we can actually investigate what genes were present in the geological past by using a comparative genomic or transcriptomic analysis. And this is actually just a very, very simple approach. What it tells us that if we want to predict the genes present in these giant trees that lived here, what we can do is we can have a look at the genes in the closest living relatives of these species. For example, in this case, plants of the genus Selaginella or Isoetes. If we look at the genes present in Selaginella and Isoetes, any genes which are found to be present in both of these lineages were likely also present in their common ancestor that lived over 375 million years ago back here, and therefore were likely also present in these giant trees. And there was one major problem though when I started my PhD and my work on this, and that was we only had omics resources for one of these groups, that plant here, Selaginella. We didn't have any for the plants Isoetes. So to start with, what we first needed to do was to go hunting Isoetes. Now, hunting Isoetes today is a lot more difficult than hunting Lycophytes in the Carboniferous. They're not giant trees. Instead, they look very similar to grass. And they're also growing in these lock environments, such as this one here in Northwest Highlands of Scotland, where they're very difficult to identify, especially when you're getting eaten alive by midges at the same time. However, I was able to collect them, able to bring them back to the lab and grow them from spores in acynic culture. And when the plants were big enough, we were able to extract RNA from their major organs. and then to sequence their genes, their transcriptome, and assemble the first de novo transcriptome for any Isoetes species containing root material. And then what we could then do is delve into the genes expressed in this plant in a bit more detail. So what I was able to do was extract tissues from the leaf, the corm, and the rootlets, the three major tissues in Isoetes. And then I was able to look at the gene expression signatures in each of these organs. And the results of doing that are shown in this principal coordinate analysis here. What you can clearly see is that the leaf samples clustered together here, the corm samples down here, and the rootlet sample over here. So first of all, this told us that there was distinct gene expression in these different organs. And the next thing it told us is that it showed us that leaves and roots had very different gene expression signatures in Isoetes. So this does not support that hypothesis that these rootlets were modified leaves. But to investigate that in more detail, what we wanted to do was to compare these genes in Isoetes with genes in other species, such as Selaginella and the model sea plant Arabidopsis, to then help us predict what genes might have been present in these giant trees, and to help us predict really if the gene expression data supported this modified shoot hypothesis or not. So the results of doing that are what I'm showing you here. So what we did is we examined the different tissues in these three different species. So for each species, we have a green icon, which represents the leaf sample, and an orange icon, which represents the root sample. And this is showing the results of looking at these on the principal component analysis, principal coordinate analysis again, for these different species. So this is showing the results on the first two coordinates, axis 1 and axis 2, which collectively make up about 80% of the variation in their gene expression. And what we can see based on this is simply that the gene expression of these different species seems to be clustering together in a species-specific manner on these first two axes. So in the case of the sea plant Arabidopsis thaliana, these two samples cluster up here. For Selaginella, we see them clustering down here, and over here we have Isoetes clustering on this side. So this tells us that the major variation in gene expression is actually between species, not between organs. However, when we looked in detail at the gene expression on principal component and axis number 3, we saw something quite different. So this axis contains 16% of the variation. And rather than seeing things clustered together by species, we now saw them clustering by organ type. So in this case, we got the Selaginella ones, these are the two squares, Arabidopsis are the two circles, and Isoetes are the two triangles. Now, what you can clearly see is that the orange samples, these are the root samples, cluster in the negative numbers on the axis number 3, and by contrast, the leaves, the green samples, cluster in the positive values. So for me, this was really exciting because it was the first line of evidence to suggest that these Isoetes roots really did look like the roots of other species, of Selaginella and Arabidopsis, and therefore didn't support this hypothesis that they were modified leaves. And furthermore, taking this lines of evidence and thinking about it a bit more in terms of the evolution of rooting systems, it allows us to predict what genes we expected to be conserved back here in these giant tree lycophytes. Because if each of these three species have some conserved root genes between them today, it suggests that these genes have been highly conserved in vascular plants and were therefore likely also present in these giant lycophyte trees that lived over 300 million years ago. So this was important because we could predict those genes, and therefore it helps support the hypothesis that these lycophyte roots really were true roots and not modified leaves as being predicted by the modified shoot hypothesis. So at this point here, we've been able to really delve into how lycophyte roots evolved, their structure evolved, and also the genes that control them. But really, the fundamental question is really then to ask, so when did roots evolve in this group? Now, to do that, we need to go back even further into the geological record and go back to the Devonian period, roughly 400 million years ago. And here's another paleo reconstruction showing what the reconstruction looked like in the Devonian period. And the red star again represents where Britain was, just a bit south of the equator. Now, in the Devonian period, for studying plants, there's one site in particular which has told us more about plant evolution in the Devonian than any other. And we're very lucky to have it here in the UK, and it's known as the Rhynie Chert, and it's located close to Aberdeen in Scotland. So here's an artist's reconstruction of what the Rhynie ecosystem looked like 407 million years ago. You can see this diversity of plants surrounding these hot spring environments, something similar to Yellowstone National Park today. Now, the Rhynie Chert itself, if you see a hand specimen of it, it doesn't look particularly exciting. So here's an example of a hand specimen of the Rhynie Chert in Oxford. However, what's really important about the Rhynie Chert is that if we take a very thin slice or a thin section through the rock, we find this structure here. So this is a thin section made through one of the plants preserved within the rock. Now, this is a transverse section again through one of those axes. And what's remarkable is despite the fact that this plant is over 400 million years old, we can see the individual cells that are making up the axis. We've got the vascular tissue at the center, we've got the cortex, the epidermis, and the cuticle, all remarkably well preserved. So the Rhynie Chert was discovered over 100 years ago, and we now have an amazing understanding of the diversity of life in this earliest preserved terrestrial ecosystem. So here's a recent reconstruction by Victor Lesky, working with Christine Strudel-Derrien to produce a new reconstruction of the ecosystem. You can see this diversity of plants, of fungi, of algae, of animals and microbes, all preserved in this very important time in the early Devonian. Now, for me, I was drawn to the Rhynie Chert, this plant here. This plant is known as Asteropsilon macchiae. And the reason why I was drawn to it is because it was a lycophyte, and it had been described as having dichotomously branching rooting axes, which you can see down here. Now, to me, these look just like the rooting axes we've been describing the whole way through this talk in the living species in those giant trees. And so I really wanted to investigate them to see if they developed that structure, that root meristem with a root cap, which I described at the beginning of the talk. So we knew that they had this dichotomously branching architecture, but in most fossils from the Devonian, you just don't have the preservation to go looking for such a minute character as this key tissue here, the root cap. But that's what makes the Rhynie Chert so special, is its exceptional preservation. So what I did is I examined thin sections of the Rhynie Chert in the collections around Britain, making the most of the amazing collections in the Hunterian, in London, in Manchester, Oxford, and St Andrews. And in total, I examined over 600 thin sections. And it was while examining these thin sections that I came across this apex shown on this slide here. Now, for those of you who haven't seen Rhynie Chert material before, just take some time to appreciate the fact we can see individual cells and cell files leading into this region called the promeristem in this apex. Now, for someone who spent quite a long time doing sections of roots, my roots of living species rarely look this good. So it really is amazing. But when I found this, there was one thing that jumped out, and that was there was no clear evidence of this root cap tissue covering the surface. It seemed to just have a kind of naked, smooth covering on the surface. So at this point, when you find one, you're never completely convinced. So it was back to those fossils to look for more evidence. And it was while doing that I came across these multiple independent lines to suggest that all of these axes, in fact, lacked a root cap. And so at this point, we're beginning to think maybe we're looking at something quite unique. These rooting axes might be just distinct from living roots. But you might be sitting there thinking, well, isn't the obvious answer here that the root cap just can't be fossilized? Well, fortunately, we were able to throw that out from work that I did in my PhD, where I focused on roots from the Carboniferous period 300 million years ago. And it was while examining those roots that I came across this structure here, which is one of the earliest examples of a fossil root meristem with root cap. And you can see the root cap is highlighted with this yellow box round the tip, on the apex of this root. So we know that root caps can be fossilized, and we know the Rhynie Chert has an exceptional preservation to fossilize them. So therefore, at this point, we began to become convinced we were looking at a new type of meristem, which had characteristics in common with roots, but also had these major differences. So what we then wanted to do was to begin to make more comparisons with these rooting axes, not just with roots, but with one of the other major meristems in plants called the shoot meristem. So I've been focusing on the base of the plant down here at the root, but at the top of the shoot, there's also a comparable structure, a comparable meristem. Now there's many differences between these meristems, but the one I'd like to draw your attention to is the structure of the surface tissue. Now in the case of roots, as we've already been talking about, they have a very rough structure known as the root cap. By contrast, the shoot has a very smooth covering of its epidermis just over the surface. So the question was, in these new examples we're finding, did they have a smooth or a rough surface? Now to do that, up to this point, we've just been using traditional light microscopy. Now that's very good at giving you the overall structure, but it doesn't give you the 3D surface of the meristems. So what we wanted to do was to image these fossils in 3D. So we turned to confocal laser scanning microscopy. This is really the workhorse of much of molecular biology today. And what we did is we put this specimen in, and we were very surprised to find that we actually got autofluorescence. So we excited with the laser, got autofluorescence, and this allowed us to see these remarkable images of these meristems. So this is the meristem here, and in this green box is zoomed up, is enlarged on this side, what we saw under the confocal. We could see all of these individual cells in the apex. And what's great about the confocal is you can take images not just in one plane, but in multiple Z planes. And in doing so, we could build up a 3D slice through this ancient fossil meristem, which is shown here. You can see the individual cells making up this apex. And in particular, this was exciting for me because I really wanted, as I said, to know whether this surface was going to be smooth or rough. So we were able to segment out the cells that make up the surface of that meristem. And in doing that, we could turn that into just a smoother visualization in Blender here. Focusing on those the blue layer here, you can see it's covered by this very, very smooth and orderly layered epidermis. So at this point, this told us that what we're looking at really was a transitional type of root meristem. It was a type of root that had characters in common with both roots and shoots. Like roots, it was below ground, it branched dichotomously, and it was carrying out the rooting function. But like a shoot, it lacked a root cap covering its apex. And so therefore it told us that roots must have evolved in a gradual fashion. So at this point here, we've been able to pin down when roots evolved in the lycophyte lineage and trace their evolutionary history through 400 million years. In the early evidence of roots, we found evidence of that highly conserved branching, and yet these were not true roots because they lacked a root cap. By the time we moved into the Carboniferous, lycophytes had exploded in their diversity. They were enormous trees, but they still had this same highly conserved architecture and root hairs. And then finally, this has been conserved all the way through to the present day today. So really, I started this talk by trying to tell you that I think that plants are often seen as a static backdrop to animal evolution. But I really hope I persuaded you that plant evolution is worth studying in its own right, and we can learn all about the fascinating changes that are underpinning these terrestrial ecosystems. So now next time you're seeing a paleo reconstruction like this, we can think of them not as simply a backdrop, but actually we can begin to understand their interesting biology, such as these early transitional root fossils underpinning the roots in the Rhynie chert, the giant roots that must have been hidden below ground of these giant trees that we can begin to understand their DNA, and finally of the roots that must have been hidden below the surface in this modern forest today. So really, plants really are the fundamental underpinnings of these terrestrial ecosystems, and they have a really interesting and complex evolutionary history. So with that, that brings me on to my acknowledgements. I need to say a huge thank you to the Professor Liam Dolan, who was my PhD and postdoc supervisor, and most of this work was done when I was still based in Oxford. And to my long-term collaborators, Hans Kerb and Chris Berry, for all their help along the way. And finally, just to say that I moved to the University of Edinburgh in October of 2020 to establish my group, the Molecular Paleobotany and Evolution Group. So if you want to learn more about the group, please follow us on Twitter and get in contact. So that just leaves me to say thank you very much for listening. I'm very happy to take any questions.